Hello students, welcome to today's Life Sciences lesson for grade 12. Today we're going to look at questions and answers based on the DNA structure from the previous lesson. So let's get going. Our first question is based on a diagram, and it says the diagram represents a portion of a nucleic acid. So there are two nucleic acids, remember. What are they? It's DNA and RNA. So we need to identify this one, right? So let's look at our diagram carefully. What can we see? We can see that you have units, we have two strands, and if we look at each unit that's in a block, you can see it's made up of three parts. You can clearly see that shape, a little circle, a little pentagon, and another little structure. And there are some letters that are used as well, right? We can see the labels V, W, X, Y, Z. And in addition, we can see the letter C that's on one of the structures on the diagram. So let's now look at the questions based on it. So the first question is, name the nucleic acid. It's DNA. Now, it doesn't tell us, give a reason for it, but I'm asking you, what allows us to identify this as DNA and not as RNA? Well, it should be obvious. It's double-stranded, and RNA is not. And this is something you would have learned in grade 10 as well, right? Let's look at our next question. Name two places in animal cells where this nucleic acid may be found. So where is DNA found in an animal cell? One should be obvious, it's in the nucleus. And what's another place it could be found in? The organelle we call the mitochondrion. Now remember, this says animal cell. If we said a plant cell, you could actually give an additional one. Which would that be? That would be a chloroplast, right? But in this particular case, animal cell, so your answers are nucleus and the mitochondrion. So let's look at the next question, which says identify. So A, what do we need to identify? It says portion V. Now look at portion V. It's in a block, and there are three parts to it. Those three parts have different shapes. What do we call that? That's a nucleotide. So remember, it's showing you one unit, monomer, but you specifically have to say nucleotide. The next one, nitrogenous base W. Now we are given one nitrogenous base, C, right? C, remember, is cytosine. So W is complementary to cytosine. So which base is that? That's guanine, right? So in a question like this, I think you should write the full name. On a diagram, we can use letters like C and G, but I think in a question where you are asked what nitrogenous base, name it, right? So write it in full, guanine. The next one, identify molecule Y, right? So molecule Y, you can see, is on the sort of backbone of your DNA strand, and you can see it's represented by a little circle. So that molecule Y is a phosphate group. The next one, identify bond Z. Right, so you can see bond Z is a bond between your nitrogenous bases. So in other words, it's helping to join the two strands of DNA together. And what is that bond? It's a weak hydrogen bond. It's fine to just say hydrogen bond, but we keep mentioning it's weak because that's relevant for things we're going to talk about later on, okay? Right, and our next question. It says, what is the natural shape of this molecule? Well, the natural shape of this molecule is not shown on the diagram. What you're seeing on the diagram is just double-stranded, but the natural shape would be a double helix, right? So remember, it would be a coiled structure. Our next question, study this diagram. So what can we see? We can see some rounded structures in orange, and we can see this purple, a double-stranded coil structure around it, and you've got labels A and B. Right, so the first question is, name the structures labeled A and B. So A, the rounded bits are histones, or you could say histone proteins. And what's B? That should be obvious. B is your DNA, right? So you know double-stranded helix, right? Double helix. Right, 2.2. Explain the function of structure A. And it says explain the function for 2 marks. Right, so let's look at your structure A. What can we see? We can see your DNA wrapped around it. So your answer would be structure A, which is your histone proteins, allows DNA to wrap around it, and this enables your DNA to fit into the nucleus of a cell. So there are 2 parts to that answer. One is structure A allows DNA to wrap around it. And the second part of your answer is this enables the DNA to fit into the nucleus of a cell. Right, so that's explanation. Right, we're stating why, you're giving a reason for whatever you stated at your first point. Right, study the stick diagram of one strand of a portion of DNA. So we usually recognize DNA in a diagram as being double-stranded. But here they are telling you, we're showing you one strand only, and it's of a portion of DNA. So what can we see? We can see single strand, and we've got nitrogenous bases projecting, and the bases are in the sequence GCTG. Right? So let's look at that again. GCTG. And then you've got a label line, and it's indicated by the letter X. Right? So look at it carefully, and let's look at our first question. The question is, complete the diagram by drawing both strands of the portion of DNA represented. Right? A drawing question. They're never complicated in a test or an exam. So there's our answer, right? And if we look at it, we can see the original strand on the left that was given in the question, and then we can see the strand you would have had to draw in, right? And what's important here is that you know how the complementary base pairing works. So complementary to G is C, complementary to C is G, complementary to T is A, and complementary to G again is C. So if you look at the sequence now, you've got double-stranded, so your complementary bases are clearly visible. There are 4 of them. What you could have added, that's a little modification on this diagram, is you could have indicated your hydrogen bonds in between, right? So if you wanted to be really accurate, you would have shown how many bonds between G and C? It would have been 3. And how many between A and T? That would have been 2. Or alternatively, sometimes we just use a colon between the complementary bases to show the hydrogen bonds, right? So that was our lesson, students. Keep answering as many questions as possible on the structure of DNA until you feel you understand it really well. So take care until I see you the next time.